Hello from Pint of Science, Armidale, Australia. But we'll kick off by just jumping straight into the rapid fire talks. Each of the students will speak for about 3 minutes, and then there'll be a bell rung at about 3 minutes to indicate that the students will wrap up. So if I could invite the first speaker, which is Lisa Huck. I hope I've presented that correctly. And Lisa will be speaking on the effects of high-intensity interval training on metabolic syndrome. So Lisa. Do you need to progress slides or just?
I'll be good. This is really nervous, like I'm really nervous. Okay, so yeah, we just start, you timing it or I just start?
Just go.
Okay.
We won't drag you off the big talk. Speak right into the mic.
Okay. High blood pressure, diabetes and cardiovascular disease are common health problems nowadays. But have you ever wondered what could be causing them? Metabolic syndrome, with prevalence rates as high as 29 to 34%, it stands as a major cause of cardiovascular disease-related events in Australia. It is a cluster of symptoms such as high blood pressure, high waist circumference, and insulin resistance, and high blood fat levels. Metabolic syndrome is seen in 70% of polycystic ovarian syndrome women, or PCOS in short. PCOS is characterized by increase in male hormones, irregular menstrual cycles, and multiple cysts in the ovaries. Both metabolic syndrome and polycystic ovarian syndrome are triggered by lack of exercises and poor dietary choices. But it is a combination of insulin resistance along with high waist circumference which worsens these two disorders and triggers an early onset of long-term health problems. Lifestyle management is the first line of treatment. So the average recommended exercise prescription, aerobic exercise prescription is 150 to 200 minutes per week. But really, how many of us can go to the gym and exercise that much? And wouldn't it be great if there was some other form of exercise that could be less intense or less time consuming, not less intense, but definitely less time consuming, and could give us a similar benefit equivalent to a 150 or 200 minute exercise? So for my PhD, I am researching on the effects of high intensity interval training, or HIIT, on metabolic syndrome. The protocol that I'm using is 17 minutes of interval training 3 times a week for 12 weeks. So what do I have for my results? With only 6 participants completed their training, I've seen a significant improvement in VO2. VO2 is the maximum oxygen consumption by the body. So you can see the first graph here. And significant improvement in VO2 means a significant improvement in cardiorespiratory fitness, which means better lung capacity, a stronger and efficient heart, and a stronger, efficient performance by your skeletal muscles, and a lot of blood and nutrition circulating in your body. The other graph here, which is of waist circumference, shows a significant reduction in waist circumference. So the fat cells which are present in our waist are directly related to insulin resistance. So increase in fat cells, more insulin resistance in the body. And if there is a way we could decrease the fat cells in our waist, there will be a reduction in insulin resistance, and that's what exactly I've seen. So as my study is still in progress, improvement in these 2 measures have indicated that HIIT is highly effective for metabolic syndrome and polycystic ovarian syndrome. It delivers superior physiological benefits, and the best of all is it's highly time efficient. Thank you.
Great job, Lisa. So our next student speaker is Richard Charlesworth, and Richard will be speaking on celiac disease diagnosis can be a pain in the arse. Richard?
So, celiac disease, or CD for short, is a chronic intestinal disorder, and it results from an immune reaction against gluten, which is a protein found in many major grains. It affects around 1 in 300 people worldwide, but this has been shown to be recently dramatically increasing in prevalence throughout the world, and it's been shown to be affecting populations which haven't classically been affected in the past. The symptoms of the condition, which include malabsorptive diarrhea, abdominal pain, skin rashes, and nutrient deficiencies, are, as you can all imagine, quite distressing for sufferers. Now, the only current form of treatment for celiac disease at the moment is a very strict, lifelong gluten-free diet. So what I'm going to talk to you about is the diagnosis of celiac disease. So at the moment, it's reliant on two main types of testing, and that's serological testing, or blood tests, and then histopathological testing, or looking at tissue biopsies. So diagnosing CD by histopathology involves taking a small sample from the small intestine and then analysing this under the microscope. Now, there are downsides with this form of testing. It relies very heavily on the actual skill of the pathologist that's looking at these samples down the microscope. And also, if there's very subtle changes in the tissue, this can be missed or misinterpreted. Misinterpreted, that was the word I was looking for. And this can lead to delayed treatment outcomes for patients. So wouldn't it be good then if there was a companion test for celiac disease, which worked on a completely unbiased and objective set of measures, where a doctor could be certain of a diagnosis of CD, even in equivocal or mild cases? So as part of my PhD, I designed a qPCR-based array. So I actually looked at the expression of 87 different celiac-specific genes at once in a patient biopsy. And I found that using mathematical and statistical modelling, I could predict the severity of celiac disease with about 96% accuracy. And I also found that I could determine celiac in patients that weren't actually presenting with it under the microscope. So I could see these changes occurring at the gene expression level before they actually happened at the tissue level. So this test is completely objective, as gene expression is either up or down, and the actual specificity of the qPCR reactions makes sure that only data from the genes of interest actually affects the predictive equations. So I was also able to show treatment progression of celiac disease, so look at treatment away from a pathological phenotype and towards a more normalised phenotype. And I'm hoping to test in the future whether I can actually use this test to separate CD patient biopsies from other intestinal pathology, particularly inflammatory bowel disease. So this work is still in the pilot stage at the moment, and it would need to undergo very large population-based testing before I could take it any further, although this was well beyond the scope of my current PhD project. So, thank you very much.
All right, and now our final research student is Chris George, and Chris is speaking on From Soil to Stress, Changes in the Gut Microbiome of Rats.
So, hello everyone, thanks for coming here and listening to my talk. So, the microbiome of the gut is a complex ecosystem of various microorganisms, which many of you may know as good bacteria and bad bacteria. Now, good bacteria, they aid in digestion, they train the immune system, and they protect against disease. Whereas bad bacteria, they can cause gastrointestinal dysfunctions and potentially lead to illness. Now, it's been found that both types of bacteria can directly communicate with the brain and alter various mood disorders such as anxiety and depression. Now, the brain's also able to communicate back with these bacteria and alter them in turn. So it's a very complex two-way interaction that we have going here. Now, following from this, the first part of my project, I will be examining how chronic early life stress will affect the gut bacteria of rats. Now, I'll be using a sort of rodent model of childhood trauma on rodents, which I will be comparing fecal samples from both stressed and unstressed rats and seeing if there's a difference. And in addition to that, I'll also be seeing if such effects have an effect on metabolism. Now, while mood can change the gut bacteria and vice versa, it's possible that probiotic treatments might help sort of balance this interaction out. And so the second part of my project, I will be testing to see if soil will have such a probiotic effect. Now, soil is a very rich source of microorganisms, and many people today feel as if we in modern society, especially children, aren't being exposed to it as much as maybe what we're used to, and this in turn is leading to all sorts of allergic effects or many other disorders. So I'll be feeding rats soil mixed with peanut butter, because how else are you going to feed it to them? And then I'll be examining if the bacteria from soil are able to permanently colonize the gut of these rodents. And from this, I will also be examining if this will have an effect on metabolism or even on the immune system. So this research on the gut microbiome and the gut-brain axis is a hot topic lately, and there's been a lot of research on it. And so with new discoveries, there's sort of the possibility that there will be new treatments available, which in the future we will maybe be able to improve our lives and our health. So thank you for listening.
The next part of the evening, we have our two main speakers. And the first of those is Dr. Gal Winter from the University of New England. So Gal is a lecturer in biomedical science in the School of Science and Technology at UNE, and she'll be talking about microbes within us. Take it away, Gal.
Thank you.
You just need to press the white button. And if you can just stand that side of the room.
Yeah, is that good? All right, thank you all. It's really nice to start the second part of the evening after we all had a few drinks, and I'm sure my talk will sound much better. So what I want to talk today about is the microbes that we have inside our body, and what exactly do they do. So we've always known that there are microbes inside our body and they support digestion and all sorts of things. But the new discovery is exactly how much microbes we have in our body. And then we also just starting to understand now exactly what they do and what they're involved in. And this is quite amazing, as we'll soon see. Another thing that we're starting to understand now is that our microbes are constantly changing, and we have the power to change them. We can influence how the microbes would change. So we have microbes inside us, quite a lot, but how much is a lot? Say a person weighs about 75 kilograms. How much of that person is microorganisms? 100 grams, 200 grams, a kilo? I mean, microbes are tiny, they don't weigh much, but you'll be surprised to know that for the average person, 2% of the body weight is microorganisms. So if a person weighs 75 kilograms, 1.5 kilograms of that would be microorganisms, which is amazing. It's trillions and trillions of microbes. In fact, it's so many microbes that it's about 10 times more microbial cells that we have in our body than human cells in our body. And if we think of our human cells and the fact that they contain our DNA, which we all consider to be the things, the thing that makes us unique, we have 10 times more microbial DNA in our body than we have human DNA. So what is really the thing that makes us unique? And if you look around you, people are different. I mean, I'm different than you, and you're different from the person sitting next to you. However, that variability between us, our DNA only encompass about 0.1% of that variability. Whereas our microbes are different about 80 to 90% between us. So that's a huge variability. So one can say our microbes are what makes us so special, so unique. You never know. And this is why we're starting to look at the microorganisms between us, within us, and consider them as our second genome. And we call that the microbiome. So that's a collective name that we have to all of the microorganisms inside our body. So where do we have microorganisms? Pretty much everywhere. We have microbes on our skin, in our lungs. Most of the microorganisms will be in the gut area. So this is where we have the most microorganisms in our body. And for that reason, every time we talk about the human microbiome, most often we actually discuss the microbiome of the gut. So everything I will show you from now on will be the gut microbiome. So microorganisms that are inside the gut. How do we get so many microbes inside us? Where do we acquire all the microorganisms? Well, when we're still at the fetus stage inside the uterus, we don't have any microorganisms. However, the minute we come out to the world, we start to acquire microorganisms. And from then on, everything we do, everything we experience, the environment we live in, will affect the microorganisms within us. So, for example, if we were born in a vaginal birth or a C-section, that will affect the microbe composition. If we are breastfed or formula fed, that will also affect the microorganism. The biggest jump in our microbial composition, and probably the most important stage, is our first years, so between up to the age of 3. This is where we acquire most of our microorganisms, and later we continue to acquire new microorganisms and encourage new population as we go through the different stages of childhood, puberty, adulthood, and aging. So we have lots of microorganisms in our body. Okay. What do they do? How are they supporting us? So if you Google this topic today, you'll find an enormous list, almost endless list of conditions that are associated with microorganisms. So I'll briefly cover a few of them. But before I'll do that, I want to talk to you about our methodology. How do we know so much about the microbiome? How do we know so much about the microbes in us? So if we look at the gut, basically once we ingest food, we start digestion in the stomach and the small intestine, and the food is later pushed through the colon, and then the end result of it, everything that is not digested, comes out as a feces. And this is where we get all this information from the feces. So this tells us what we have inside. So basically what we do to learn about this stuff is we analyze feces sample. So if you're thinking, wow, you know, this woman, like me, I have such an interesting job, just remember that this is what we do. And this is what it takes to get there. We analyze the feces, and we look at which microbes are inside the feces. Now, a lot of the studies are what we call link by association. So, for example, you will study the microbial population in healthy individuals, and you will study the microbial population in diseased individuals and compare between them. And then you'll say, oh, all the disease have these and these and these microbes, and therefore these microbes are connected to this disease. This is what we call link by association. You can take it one step forward and establish what we call a causal relationship. And this is with the use of fecal transplantation. And yes, this is exactly what it sounds. You take the feces from one, let's say, organism, and you transfer it to a different organism. And then you study the effect. And if by transferring the feces from a diseased individual to a healthy individual, they develop the disease, you can say, there must be something inside that feces. There must be microbes inside that feces that are causing this disease. Does that make sense? Very good. So I'll give you some examples of these sort of studies. And this first one is about obesity. And in this study, they try to establish a causal relationship between the microorganisms in the gut and obesity. And they used for this study, they used 4 pairs of female twins, so human female twins, which happened to have one obese twin and one lean twin. Still with me? Good. All right, so they isolated feces from all subjects, and then they transformed it, injected it into mice. And what do you know? The mice that received the obese microbiota became obese. And the mice that received the lean microbiota, they stayed lean. So this goes to show you a clear cause and effect. Because they received the different microbiota, some of them developed obesity and some stayed lean. Now, in that particular study that was actually published in Science magazine, which I don't know if you're familiar with, but is one of the most regarded science magazines, they took another step, and after the mice were established, they tried to transform the microbiota in between them. So to give lean microbiota to the obese mice and vice versa. And what they found is that if you transfer the lean microbiota to the obese mice, they rejected it. It just wasn't accepted. But if you transfer the obese microbiota to the lean mice, then they accepted it. So this will go to show you about the cause and effect relationship, and also the relationship that our diet and the gut environment has in accepting new species and creating changes. All right, another example that I want to show you is something that Chris has talked about earlier, and it's this new and very exciting area that is called gut feeling. So when I first learned about it, I was blown away. So as we discovered today, there is a clear connection between our gut and our brain. So processes that are happening in our gut can affect our brain, and vice versa. If we're stressed, or if we're depressed, or on the other hand, if we're super happy, that will also affect our gut. And this is generally coined as gut feeling. So when I first learned about it, I was shocked. How how can the microbes in our gut get all the way up to our brain? I mean, there's quite a huge distance between the two. But there are several communication pathways between the gut and the brain. One communication pathway is, as it turns out, we actually produce a lot of chemicals in our gut that could be active inside our brain, like serotonin, dopamine, and other neurotransmitters, if you're familiar with that. These chemicals can transfer from our gut to the brain. Now, the population of microbes that we have in our gut can affect how much of these neurotransmitters we produce. And then that can affect our brain. And moreover, our gut population can also produce these these neurotransmitters. So bacteria can produce serotonin, dopamine, and all sorts of neurotransmitters, and then these neurotransmitters can then transfer to the brain. Another pathway of communication is microbes can directly or indirectly stimulate the nerve that goes from our gut to the brain, and also the other way around. Processes that are happening inside our brain can transfer to the gut and modulate our microbial population in the gut. And of course, I didn't mention communication with the immune system. So our immune system is in constant touch with our microorganisms. And the identity of the microorganisms that we have in a gut can trigger the immune system and get it to alter brain processes. So there are many, many ways of communication. At this stage, we don't really know. We can't say this is happening when you're feeling depressed, this is happening when you're sad, but this is where research is heading, to identify these processes better. Now, I'll quickly go through the other examples, but we know that the gut microbiota, the microorganisms in our gut, can affect the way that we metabolize food. So different people will have different responses to different foods. Some will get really fat, some will stay lean, and the gut microbiota has lots to do with it. Gut microbiota is also affecting how we age. So this is a nice experiment where they used fish, and they injected an old fish with a young fish microbiota. And what do you know? The young fish live longer. So maybe, like 20, 30 years from now, we won't even need to do facelifts and all this stuff. We'll just take microbes. Gut microbiota also affect different bowel disease, like inflammatory bowel disease and Crohn's disease. It affects cardiovascular disease because it affects the way that we process fat and the accumulation of fats in the arteries. And it affects autoimmune diseases because of the connection between the immune system and the gut microbiome. Now, I don't have a slide for it, but it also affects how sensitive we are to different allergies. I mean, have you noticed how many allergies are around us? Gluten. Gluten intolerance, lactose intolerance. So gut microbiota play a role in this as well. And it also is associated with cancer disease. So pretty much, and this is just a partial list. If you look it up on Google, you'll find many, many other conditions that are associated with the gut microbiota. So why is that? Why is everything so associated with the gut microbiota? And why is the gut microbiome so different between all of us? So microorganisms, as I'm sure you all know, microorganisms are very responsive to the environment, and they're very adapting to the environment. And if we think of our gut microorganisms, their environment is our gut. So we have total control of their environment. Probably the most important thing is we need to feed them. We need to feed the microorganisms inside us. And they eat what we eat. I'll just keep talking. So if we eat a certain food that would only feed a certain group of microbiomes, we would give this group of bacteria a huge advantage because we're giving them more food to live and prosper. If we have a certain group of microbes that requires a different type of food, which we don't eat because, I don't know, maybe we don't like it, maybe it's not available, then we practically eliminate the existence of this microbial group. Oh? All right. I hope you got a good look at the slide before it went away, because it had all-you-can-eat gut buffet. And the point of it was to demonstrate that we feed the microbiome, we modulate the environment, everything that we do, everything that we change in our body would be changing in their homes, changing in their environment. If we exercise, if we smoke, if we're stressed, if we're happy, every little thing would affect their environment, and microbes will respond to that. All right, so, well, I think I've pretty much covered that. All right, so you probably ask yourself now, oh, wow, well, microbiome is really important, and I want to get myself a good one. How can I do that? And the answer is that we don't know. So at this stage, we can't say, do this, this, and this, and you'll have a good and healthy microbiome. We don't know that much yet. What we do know, and what is becoming very, very clear in all of the studies, is that the more microbes that you have, and the more types of microbes that you have, the better. Okay, so every disease is characterized by having less microbes and less type of microbes. So if we want to do something about it, the best thing we can do is to keep the diversity. Have as many microbes as we can. And how can we do that? Well, by diversifying our food. For example, if we eat lots of different nutrients, we are likely to feed lots of groups of microbes. And the other thing is, we need to increase our exposure to microorganisms. And this goes to what is called today as the hygiene hypothesis. We are way too clean. We don't need to be this clean. We need to have microorganisms inside us. So dirt is quite good for us, especially for young kids, but also for adults. So the more we're exposed to dirt, the more we're exposed to microorganisms, the more diverse our microbiome is, which makes us stronger. So if I can, live with one good advice, it'll be get dirty. And that's it.
That was a great presentation. And look, I should mention that after the presentations, we will have the speakers up in front, and we'll have a question and answer session. So if you've got any burning questions, you'll have a chance to throw them at the speakers. So our next speaker is Dr. Robert Hart from the University of New England, complete with cheer squad. And Rob is also a lecturer in biomedical science in the School of Science and Technology, and he'll be talking about amazing appetites.
Yep. Cheers. Okay, that, I think. All right, thanks for having me. Nice to see everyone here. Getting a dose of science, I hope. I'm hoping that I might be able to bring things together a little bit with my short talk. So my area of research has been looking at a hormone which is related to regulating appetite. And so what I did was, for my research, through my PhD in particular, was I would inject animals with this hormone and I would try to find where it went throughout the body. And so just to get an idea of what the hormone is, it's leptin. So I work in the same lab as Eliza does. And basically, the way that leptin is thought to regulate appetite is that it signals back to the brain, giving a signal saying that you've got enough energy available and you don't need to eat anymore. And so the way that appetite is more broadly regulated is that you have signals like this, hormonal signals, which are secreted from the digestive tract. They get into the circulation and ultimately get to the brain, and they cause this signal, and you stop eating. You also have other signals, like actual physical signals coming from the gut. So as you start eating, your stomach starts to expand, and you have, say, the vagus nerve, for example, sends a signal saying that your stomach is so stretched, you don't need any more food, you stop eating. And so by having these signals, basically you limit food intake. So if you don't have these signals, you might see something like Prader-Willi syndrome, where people just continue to eat and overfill because they don't get that signal saying that there's enough food in their body already. So what we see is that as your stomach starts to then push the food into the small intestine, stretch signal starts to dissipate, and you also find that you start getting different hormones being secreted. So you start losing the secretion of leptin, you might get the secretion of, say, something like ghrelin, and so consequently over time you end up with these signals going back to the brain saying, okay, I'm starting to get hungry again. And so you start seeking out food and looking for something which is going to fill you up and keep your body working, hopefully, give you enough fuel so that you have your normal metabolic processes just occurring as you would expect. So how does this go wrong? And I'll end up in things like obesity, which is one of the areas that I'm interested in looking at. So I think everyone probably knows obesity is just an excess of fat. So we have a definition of basically, what is it? Height squared divided by mass, and you work out your body mass index, and anything greater than 30, you have an obese body mass index. And so generally you'd see that you have this accumulation of excess fat, and that's because fat is the most efficient way for the body to store energy. Fat is the most energy-dense product that you find anywhere throughout your body. So if you've got lots of energy and you need to store it, that's where it goes, into fat. So this excess storage of fat occurs as you find that energy balance gets thrown out of normal balance. And so you end up that with, you have more energy coming into your body than what is actually expended. And so this excess energy starts to get then put away so that the body can use it later on, because basically we've evolved through time to actually be able to adapt to starvation situations. And so that means that our bodies are really well equipped to put this excess energy away so that we can use it later on as we are evolved to actually do. You know, once you've got to a famine, you need to use that fat so that you can keep living, basically, and start finding other food sources by using that energy. And so what we find now with what you might see referred to as the Western diet, like you see in that figure there, is that we have highly palatable food, which is highly abundant and very easy to get a hold of. And so this has way more energy than we'd normally expect to expend in a single day. And so you end up with this energy surplus, and so you end up with that fat being stored throughout the body. And so ultimately you end up putting on weight. So the way that this would occur is that, so I talked about how the appetite is actually regulated. You normally have what's a negative feedback loop. It's basically you've got enough of whatever it is, in this case food, signals to the brain, and you get a stop signal. So in this case, stop eating or stop seeking food. And so as you have this excess of fat starting to build up in the body, you have an excess of that hormone that I was talking about, which is leptin, which is normally considered to be a stop signal for that food-seeking behaviour. So as you have this higher concentration of the hormone, you find that the body basically sets a new set point and becomes less sensitive to that signal. So it's basically saying that there's a threshold here, which is normally where you'd stop eating, but in this case, because there's that much leptin in the circulation normally, the threshold is pushed higher. So you have to secrete more leptin to get that fullness sensation. And the only way that the body can do that is to produce more fat. So I don't know if I mentioned before that leptin is actually secreted by fat. So to actually get that signal so that you stop eating, you have to put down more fat so that you can produce more leptin so that you get that fullness signal. So it's a pretty horrible positive feedback loop that we talk about in this case, where you have something which starts to spiral out of control. And one of the consequences of that is that leptin also interferes with other signals. So it uses up other intracellular signals, intracellular proteins, which it uses to signal. So it interferes with signals coming from other hormones like insulin. So you see things like diabetes are often seen in people who are also obese. So you get this whole crossover of all these metabolic diseases. So I think Gal mentioned metabolic syndrome before. And so that's one of the ways in which you would normally expect that to start to occur. So one example of this would be where we've had genetic mutant mice have occurred. And you can actually see that we've got one of the mutants at the top of the picture there and a lean littermate for comparison next to it. And what we see in these mice is that the one that I'm talking about at the top there is actually insensitive to leptin at all. So it lacks the normal receptor, which would say, for the hormones to actually work, you have to have them get into circulation, and then they bind to what's called a receptor, which only recognises that hormone. And that's what actually gets that signal into the brain. And so these mutants don't have that receptor, so they can't receive that signal. So this is similar to what you see with that Prader-Willi syndrome, where they've got this insatiable appetite. And they actually have a much higher concentration of leptin in circulation because the body's there just producing more of it to try to get this signal, which never actually gets through. And so you have this really horrible spiral out of control. And you see that in these animals, you also get high insulin concentration, high glucose concentration in their plasma, so in their blood. And so that's characteristic of diabetes. So that's basically metabolic syndrome. This is a good model of metabolic syndrome. But what we also see is that these animals, compared with their lean littermates, have this vastly different gut microbiota, which Gal was talking about. So I'm hoping that maybe I can tie this back together a little bit here, in that what we see in both animal and human models of obesity is that that gut microbiota seems to be characteristically different between both lean and obese subjects. And so what Gal was talking about before was that these microbes are actually in your guts, and they're producing a whole series of different chemicals which are structurally very similar to what you find in the body being produced already. And so that means that the products that are being produced by these microbes have the possibility of getting out into the body and causing disruption of the normal signaling that we have. And so what it seems to be is that there are certain microbes which lend people to be more susceptible to become obese and develop this metabolic syndrome. And you see a very characteristic profile where these very particular species are found at a higher, found in a higher proportion compared with the other normal species between obesity and lean subjects. And you also see that through other conditions, so depression or anxiety. It seems that these models also have very characteristic profiles in their gut microbiome makeup compared with normal controls. So that's one of the main reasons why we're starting to see that there's this huge explosion in the actual research going on into the gut microbiota. One of the other things that we see is that the so-called healthy or the slimmer models have much broader diversity in the gut microbiome. So there's a much larger number of different types of species in the gut. And it seems that by having this diverse microbiome makeup, it makes the individuals more robust and more able to cope with any stresses that might disrupt their normal healthy state of being. And so, like Gal was saying, it seems that by having this broader diversity, you're more likely to have a healthier, more likely to be quite healthy by comparison with anything where you've got a more narrow number of species. And we're starting to see actually, I think in the US, they've got data on fecal samples for people for 7 generations. And what they're finding is, as they're going through these generations, there's actually a narrowing in the diversity. And so we're starting to see that as it seems, I don't know, if I feel like this is maybe going into that hygiene hypothesis. It seems like maybe we're cleaning up our environment so much that we're becoming less exposed to new microbes. And so it seems that we're making it more likely that we're going to develop one of these chronic conditions because we're not exposing ourselves to this broad range of microbes, which is actually beneficial to our health. And so, like Gal was saying, as you eat, you're feeding your gut microbiome, and we want a variety of those microbes. So we need a variety of fuels to actually fuel, to allow these different microbes to eat different foods, basically. So what we recommend is a diverse diet. Make sure that you feed all of these different microbes. And one of the more interesting things that I came across was that people who had at least 1 drink a week seem to have an even more diverse gut microbiome in comparison with people who didn't drink at all. And so, based on that, I think we can justify having 1 beer while we're here tonight. So, I'll just leave it at that. I hope everyone's had a good night. Thank you.
Does anyone want to kick off with a question for any of our speakers over here in the front?
What is the best thing to ingest to increase your microbiome? After soil? Well, I have an answer, but it's kind of gross.
Sorry, can we just get you to repeat the question just in case others didn't hear it?
After soil, what is the best thing for one to ingest to increase one's microbia? So I said my answer is kind of gross.
But you can elaborate, go
on. So I was thinking, fecal transplantation. But actually, I just recently read an article that said we should pick our noses more. And we'll get lots of microbes in there and also lots of immune factors. But I think to put it into real life answer, it would be the diversity of food and also keeping a healthy environment. So like exercising and keep changing the colonic environment.
All right. Another question? I have one. So we hear lots about probiotics. Can you give us an overview of the value of probiotics, if there are any out there that really do good things? Or?
Well, this is a bit tricky because there's something tricky about probiotics. I think it's a bit more of a marketing term than true science, but not really. So there are evidence of probiotics being helpful and supportive in digestion processes, and there are evidence that showing that if you ingest probiotics, some of the microbes that you ingest can compensate for metabolic activity that you may be lacking in your own body. The thing with probiotic research today is that we don't really know how long these so-called good bacteria are, how long they stay inside the body. So most of the studies that have been done, they looked at, let's say, probiotic supplementation for about a month, and they checked the body activity for this month and nothing after. So these studies definitely hold, but I don't know, there isn't much data about the long term of probiotics.
So, another question over here.
Rob, you talked about digestion, appetite being suppressed and that sort of thing. When you fall ill, you want to stop eating for maybe a couple of days even. Does that have to do with the microbes as well?
I think there's a portion of that, and I think it's also part of the inflammation that you see throughout the body. So you see when you become ill, you have a whole series of reactions occur, and you have basically your whole immune system becomes active to try to fight off whatever pathogen it is, so whatever problem it is causing the disease or sickness at the time. And so part of that does seem to be that, so initially you might have the hypothalamus, part of the brain gets affected, and that raises the temperature. So you start to get a fever, and that part of the brain is also very close to the same part which regulates appetite. And so I think that it's probably more to do with the inflammation reaction that you'd see. And I suspect that by having that message going to the brain, then you might get another message going back to the gut, and I think that would then cause a change in the gut microbiota. So I suspect it's probably more to do with the immune system actually becoming active and having an effect at the brain rather than the gut microbiota potentially being affected themselves. Certainly if it's, say, a gastrointestinal problem, then definitely you'd see that there's a direct effect on the gut microbiota because whatever microbes have been introduced, they're going to be then competing with the microbes which are already in your gut. So it's basically an ecosystem which has been thrown out of balance by this new species being introduced. And so potentially that does have an effect, but I think that the actual effect that you're seeing is more mediated by the immune system rather than by the microbes directly. But unfortunately it's not really my area, but that's my feeling. No worries.
So I'm going to ask a question of each of the students. So, look, I'd like each of you, and you don't have to, elaborate too much if you don't wish, but can you tell us why you got interested in science?
I'll go first, and these guys can follow. I've always been fascinated how things work. And like the intricate detail of the body, how everything's so tightly controlled but sometimes gets really out of whack. And I just have always liked science. I'm really lucky, like I did my biomed degree and I did honours, and now I'm doing medicine, so I'm going to learn everything about the body, which is apparently what I wanted to do since I was like 12, so really lucky to do that. But I'm particularly interested in reproductive physiology and fertility because I think science has been around for such a long time and studying systems has been around for hundreds of years, and I think some areas have been maybe sometimes neglected, and I'm really excited to be part of contributing to research in these kind of areas as well.
Great.
I was sort of set up from a pretty young age with family members just giving me encyclopedias before I could actually read. Although my first choice for a career was actually paleontology, but I couldn't really see that being able to support me in the future. Yeah. I mean, I do read into that a lot just as sort of a hobby because I'm a filthy nerd. But but in what I'm doing now, after high school, I was just reading through the university options and I thought, oh, this seems interesting. I'll just go with that. And now I'm here.
As you do. Next.
So, same as what Eliza said, I've always been fascinated by a young age at how things work. I was always that kid that would take things apart and mostly put them back together again. So I got into immunology in probably my last year of university. And at first it was one of those very daunting subjects that you go to and no one understood what was happening. We were all very, very confused. But it was like learning a language after a while. And once you start to sort of speak what's happening with immunology, it all started to make more sense. So yeah, that's sort of the field that I've gotten myself into.
For me, science started when I was a little kid. So I remember my sister and I would play together and we would name the plants with different medicines. So I still remember I would call one of the rose bush as Dr. Soframycin. so yeah, and I took that journey towards medicine and I did not succeed. So I did physiotherapy. So I'm a physiotherapist by profession and then I had 3 kids and I lost it all. Like I put on weight and everything and I'm like, okay, how do I change all this? And the only thing I could change everything and myself with was movement. So movement physiology interested me. And that's how I landed in exercise physiology. I did my honors with polycystic ovarian syndrome and now elaborated my research to metabolic syndrome. It's just because of movement and how movement can affect the entire body and change the total physiology. Yep.
Great. So we have a question up at this table and then one down here.
This one's for Speakinum too. Sorry, I'm not good with names. With your method for how you actually did, like you were sequencing the the genetic material for the Crohn's disease, are you able to adapt it to other diseases as well?
So what I was looking at with celiac disease was actually the messenger RNA. So when you have a signal from your nucleus and it gets transcribed, you get messenger RNA. So it's a way to sort of get a snapshot of what's actually happening in the cell before you get to the protein level. So a lot of the genes that I looked at were genes that are involved in very common inflammatory pathways. So there are certainly pathways that sort of overlap between Crohn's disease and celiac disease, but there are pathways that are certainly more specific to Crohn's disease. So that's what I'm hoping to look at in the future. Does that answer your question? Good.
And we had another question down the front here.
Thank you. From what I can gather, the body has a pretty good idea at any point in time as to what it thinks a reasonable appetite is. And you and you mentioned the cycle where it keeps going up. I seem to remember a large study of a large number of people, different diets, and after 5 years they came to the conclusion that the results for all of them were 0. I don't know if I got that right. I probably got it wrong. Which seems to imply that there's a method for the body to think a bigger appetite is better, but there's not one for bringing it down. Is that correct?
I certainly think that's the case. Like I was saying before, we seem to be well adapted for famine conditions, not for feast conditions, and so our bodies seem to be genetically programmed to want more energy, and so we've got methods to store that excess energy, but there are less methods that our body has to actually utilise those stores, because we're basically programmed to become ready for this impending famine, which has always been there as a natural selection pressure throughout our evolution. And so it seems that we've evolved to get these signals that we always have this stimulation to go and seek more energy, more food, as we start using whatever energy we have in our body. And so that seems to have resulted in where we've got this excessive energy available in the environment now, we can just keep gorging ourselves, and so we lose the sensitivity to that signal, basically saying that we're full. And so that means that the body, as it loses sensitivity, we need a much bigger signal to get that message back in, to actually get to the point where we feel full. And so that's basically the hypothesis, which is leading to obesity, in that, say, if we're just talking about leptin, there's what we call a leptin insensitivity. Just because we lose that ability to receive the signal, we need to get a much bigger signal to get that, much bigger stimulus to get that signal to be received. So we have to produce more of that hormone, and to actually get that to occur, we have to have more fat available to produce that signal. It seems that there are some ways to bring that back down, but it's a lot harder than raising that set point. And so certainly gastric bands seem to have some effect, but not quite as good as gastric bypass surgery, where you actually physically staple off part of the stomach. It seems like the stomach itself is actually secreting a number of these hormones which are stimulating appetite. And so by restricting that bit of the body, it means there's less of the hormones getting into circulation. And so that's one of the reasons why I think it's ruined why gastric bypass surgery seems to be so effective. It actually changes the hormonal profile in individuals rather than just physically limiting food intake. Does that answer it? Yeah.
All right. Look, we don't have a lot of time.
There's one more.
But we'll take one more, and I'll take guidance from the organisers as to how long we keep going for.
So the question I'd like to ask is if you can give insulin to someone who's got insulin insensitivity, like a diabetic patient, why can't you just give leptin to someone with leptin insufficiency? Seems to make so much sense to me.
Well, you can to a degree, but again, you have to raise that level so much further above than what you normally have. So to actually produce that insulin, what normally you'd see in a factory is that you have a whole series of bacteria which have the gene for insulin inserted into them, and they'll actually produce that insulin, and that has to get harvested. And it doesn't seem, at this point anyway, that it's viable to do that with other proteins like leptin.
Also types of diabetes, so juvenile
right,
onset where you lack insulin, and then mature onset where you're insensitive to insulin. Yeah. Question over here? Yeah. I'm happy to let it run, doesn't worry.
Yeah. See how we fare.
There are a lot of things I've read about the values of fasting. For example, the 5-2 fast, and people who will even fast for a week or 2 weeks and say they feel a lot healthier. How does fasting affect either the leptin or the microbes or something? You know, what is going on that makes people feel better?
Certainly for leptin, the concentration plummets, and so that stimulates that food-seeking behaviour as you go into a fast situation. So it seems like as that leptin concentration drops, it's signalling to the brain that you're going into a fasting situation, so you need to go and seek more food. I haven't looked into what it does to the gut microbiome, but I have read some recent research which suggests that the 5-2 diet isn't actually any better than, say, caloric restriction. It seems like they both have a similar effect on weight loss, assuming that's the goal behind the diet. So if that's what you're actually after, weight loss, it actually looked as though, from the data I was looking at, it looked as though the caloric restriction was a bit harder to get used to for the subjects. But once they got used to it, I think it looks like they took about 3 months to get used to it. They were better at sticking to that regime than the 5-2. 5 days eating, 2 days fasting, or restricted intake treatment. So it seems like if you were to go for a long-term treatment, probably caloric restriction would have a better outcome based on those data. And that was a pretty large study. I think it was several tens of thousands of people in it. So I'd be pretty confident that those data were reasonably good.
So do we have any more questions? Another one up the back and then one down the front here. And you're not going... All right. Last two, and then we'll draw it to a close. The publican's starting to come up the stairs, I think.
A question for Chris or maybe Gal. Have you tried eating the dirt yourself?
Chris. It's got peanut butter in it.
I don't actually like peanut butter, so.
Tried Nutella as well.
Yes. We're testing to see whether the rats would like Nutella or peanut butter more.
Boiled up out of the thing.
They haven't boiled up, they just sort of went a bit funny. We had to sterilize it first. Nutella separates a bit. But no, haven't tried eating it.
All right. So one last question down the front, and then we have one more duty to perform, and then I think we're about done.
Hi. Okay. My question is directed towards Ms. Hark. Your studies into high-intensity training, have you found that to be more effective than regulating your food intake and controlling your weight?
I haven't seen a lot of weight change in my participants for the 6 people who have finished, but I'm not looking at diet at all. So I do not tell them what to eat, what they should not eat. It's up to them. All they do is come and exercise for 17 minutes, 3 times a week for 12 weeks and go. So, and that's the result is what I've told is like an improvement, like reduction in the waist circumference. But I haven't seen, like, their weight has gone down slightly, but not, like, I could not bring it up here on my slide because it was not significantly lower. And when we research, we need to talk at, like, with statistical significance. So, yeah, but the weight definitely, I feel is like, if you're eating a lot of sweet food, burgers, donuts, and all that, the picture that Rob showed, you won't see a weight change ever, ever. In fact, you'll see your weight going up all the time, however much you exercise. So the concept like, okay, I'm eating a donut or maybe 5 donuts and then I'll go and exercise. You can't do that. It does, yeah, you have to. There has to be a balance with everything, yes. But, yeah. Yeah, but physiological benefits of HIIT are enormous. The biggest of all is like you can finish it off in a tiny amount of time compared to like a 35 to 40 minutes of moderate intensity exercise that you go and run, whereas this is over in just 17 minutes. Yeah.
We'll have to draw the questions to a close there, but look, I'd like you to thank all of our speakers tonight, Gal, Rob, Lisa, Richard, Eliza and Chris. So if we could just put our hands together. And so the last task that we have tonight is to present a prize for the rapid fire winner for Monday and Tuesday nights. And the prize is an iPad donated by the UNE School of Science and Technology. And the UNESTA people have told me that the winner is Richard Charlesworth. So, did you want to say something, Richard? All right, great. And I hope everybody here has also had a fun night. And so we should acknowledge, as we can see on the board here, the suppliers of the prizes: UNESTA, UNE, Ecological, Cyro, Costa Tomato Exchange, Domino's Pizza, and Bikram Yoga, New England. So finally, I have to give special thanks again to our speakers, Gal and Rob, our rapid fire student speakers. Our sponsors, Cyro, UNE Science and Technology, Inspiring Australia, New England Northwest Regional Science Hub and Ecological. Our venue, the Wicklow Hotel, for kindly hosting the event, and the UNESTA volunteers who've worked tirelessly to make this event happen in Armidale, and they have really done a great job. And I think the event's run, at least in the first 2 nights, really smoothly. I know last night there was terrific feedback, and it's been a pretty pleasurable night, I think, for everybody here. So thank you. Thank you, everybody else, for attending. It's been a good evening, and safe trip home. Enjoy the rest of your night. Cheers.